Imagine being able to completely rewrite an animal's social life. And I mean, not with drugs, not by, shining lasers into its brain, but physically installing this microscopic lock and key electrical bridge that was actually taken from the eye of a fish.
I know, it sounds completely like science fiction, right? But it is this massive, massive leap forward in neurobiology. We're looking at a totally new way to structurally edit the mammalian brain.
Exactly. And that is why we are taking you on a really highly detailed journey today. So welcome to the deep dive. Our mission today for you curious learners out there is to unpack this incredible, groundbreaking Nature paper. It details a brand new technology called LinCx.
Right, which stands for a long-term integration of circuits using connexins.
Yeah, LinCx. Think of these scientists as like master electricians. They basically figured out how to install these custom, highly secure cables between specific brain cells to actually alter behavior. And they do it using the brain's own natural power grid.
Which is just wild.
It is. So, okay, let's unpack this. How on earth did a protein from a white perch fish end up changing stress and social behaviors in mice?
Well, to really grasp the magnitude of this, we first have to zoom way, way in on how brains actually communicate. Because usually when you hear about brain signals, you probably think of chemical communication.
Right, like dopamine or serotonin.
Exactly. A neuron releases a chemical neurotransmitter, it floats across this physical gap, and it lands on a receptor. But today, we are entirely focused on the brain's other communication network, which is direct electrical signals. We are talking about electrical synapses.
And historically, trying to precisely edit these electrical synapses in a complex mammalian brain has been, well, it's been like trying to rewire a single room by flooding the entire city's power grid. It's just chaotic.
Complete chaos. And to understand why it's so chaotic, we need to meticulously break down the actual physical anatomy of one of these brain wires. So picture the outer membrane of a single brain cell.
Okay, got it.
Embedded right in that membrane is a structure called a hemichannel. That hemichannel is an oligomer.
Which means it's made of multiple parts, right?
Yeah, it's a complex biological structure built from 6 individual proteins, and these specific proteins are called connexins.
So 6 of these connexin proteins wedged together, almost like, I don't know, slices of a pie.
Yeah.
And they form one half of a tunnel, the hemichannel.
Precisely. Now, to make a complete electrical synapse, which we call a gap junction in the field, 2 of these hemichannels, one from each touching cell, have to perfectly align and physically dock together.
They just click together.
Right. They fuse to create this secure tunnel. And that tunnel allows ions and small electrical currents to flow directly from the inside of one cell to the inside of the other. It's physical. And mammals, including humans, we actually have 21 different variations or isoforms of these connexin proteins.
But there is a massive evolutionary catch here, right? Like a structural design flaw when it comes to us trying to engineer these circuits.
Yeah, absolutely. Most mammalian connexins form what we call homotypic junctions. They strongly prefer to dock only with identical hemichannels. So like a connexin 36 hemichannel reaches out, and it will only dock with another connexin 36 hemichannel.
Which brings us to this fascinating warning case mentioned in the research involving C. elegans.
Oh, the worms. Yes.
Yeah, these microscopic worms. Scientists previously tried to take vertebrate connexin 36 or C-guy 36 and put it into these worms to rewire their olfactory, their smell circuits.
And worms are a great testing ground for this because they don't naturally have connexin proteins at all. They evolved a totally different set of proteins for this job. So when researchers introduced mammalian C-by36 to connect 2 different types of worm neurons, it worked beautifully.
Because it had nothing else to accidentally bump into.
Exactly. But when they tried to express that exact same C-by36 in 2 sensory neurons of the same cell type, the whole system just broke down. The otherwise normal worms showed completely disrupted, chaotic behavior in response to smells.
And that's in a tiny worm with exactly 302 neurons. I mean, mammalian brains are infinitely more complex. We have millions of the exact same cell types packed tightly together in these really dense circuits.
So you can see the problem here. Because vertebrate brains are packed with millions of identical cells, introducing a new homotypic connexin to connect cell A to cell B is, well, it's impossible to control.
That's just going to connect to everything.
Right. That homotypic connexin will inadvertently create off-target electrical synapses between all the nearby cells that are naturally expressing it. You essentially lose all anatomical precision.
It's like giving everyone in a massive crowded city the exact same walkie-talkie frequency.
Oh, that's a good way to put it.
You don't get a private, secure line from point A to point B. You just create this chaotic, citywide feedback loop where everyone is shouting over everyone else.
Mhm.
So to do this right, we need a walkie-talkie that only has a transmitter on one end and a receiver on the other, right? A totally exclusive channel.
That is the perfect analogy, and it highlights exactly what researchers needed to find. To precisely edit a brain circuit without causing a massive short circuit, we needed a heterotypic connection, a lock and a key that only fit each other and completely ignore the billions of other connections in the mammalian brain.
Okay, so let's do a quick recap for you listening before we move on. Number 1, electrical synapses are built from these connexin hemichannels. Number 2, normal mammalian connexins dock with themselves. They're homotypic.
Right.
And number 3, this causes chaotic off-target wiring in complex brains. So a question for you to think about. Think about the communication networks in your own life. What happens when a message meant for one specific person accidentally goes to a massive group chat? Total chaos, right?
Absolutely.
So if I'm tracking this right, mammalian brain wires naturally dock with themselves, which causes a tangled mess. We needed a completely foreign lock and key, which means we had to look entirely outside of mammals to find a clean wire.
Exactly. They looked to the water, specifically the white perch fish, Morone americana.
Morone americana.
Yeah. This fish expresses two highly unique homologs of the mammalian CGI36 protein. They are called CI34.7 and CBI35.
So what makes these two specific fish proteins the holy grail for brain engineers? Why these ones?
Well, they naturally form a robust heterotypic gap junction, meaning CGI34.7 strongly prefers to dock with its partner, CGI35. Furthermore, this specific pairing has an inherent directional flow.
Wait. Directional.
Yeah, the electrical current actually prefers to travel one way from the CI34.7 side over to the CBI35 side. It acts just like a diode in electronics.
Okay, that sounds ideal, but I have to play devil's advocate here for a second. If you take fish proteins and just drop them into a dense mouse brain, or eventually a human brain, how do you guarantee they won't just accidentally dock with the mouse's own native proteins? We don't want cross-species walkie-talkie interference.
And that was the defining engineering challenge of this entire project. They had to guarantee absolute exclusivity. They needed to mutate these fish proteins to make them structurally invisible to mammalian connexins.
Wow, invisible.
Yeah. And to do that, they needed a way to test millions of mutations incredibly fast. So they utilized a brilliant in vitro assay called FTC.
FTCH. That stands for Flow-Enabled Tracking of Connexosomes. I really love the mechanics of this test. Let's use some visual memory here to paint a picture for the listeners of how researchers actually proved two microscopic cells have successfully docked.
Okay, so picture two separate populations of human kidney cells floating in a dish. These are HEK293FT cells, very standard for lab testing. You genetically engineer one population to produce your lock connexin, and you tag them to glow bright green using an emerald tag.
Okay, bright green lock cell.
Right. Then you engineer the other population to produce your key connexin, and you tag them to glow deep red using an IRFP670 tag.
So we have a glowing green pool and a glowing red pool.
Exactly. Now you mix them together. If the two different hemichannels successfully recognize each other and dock, a fascinating natural cellular process actually takes over.
This is the part that blew my mind.
Yeah, the fully docked gap junction is eventually swallowed whole by one of the two cells, forming a tiny double bilayer bubble inside the cell.
And that swallow bubble is what we call a connexosome.
Exactly, a connexosome. And think about the visual result of that process. If they dock, the red cell literally swallows a piece of the green cell, or vice versa. The cells swap colors.
These swap colors, that is wild.
Yeah. The scientists then run these millions of cells through a flow cytometer. That's a machine that uses lasers to rapidly count and analyze individual cells based on their light emission. If the machine detects a high number of individual cells glowing both red and green, you have definitive, undeniable proof of physical docking.
That is just incredibly elegant.
Right.
But I want to dig into the actual mutation process for a minute. They didn't just, guess and check. They fundamentally altered the physical anatomy of these proteins. How exactly did they redesign the lock and the key?
Well, they used advanced computer simulations, specifically a technique called homology modeling, to map the exact atomic structure of these fish proteins, and they focused their attention entirely on the extracellular loops known as EL1 and EL2.
EL1 and EL2.
Right. Think of these loops as the physical hands of the protein that actually reach out beyond the cell membrane to grab the other cell.
So if the handshake matches, they dock.
Precisely. The researchers painstakingly tweaked the specific amino acids that make up those loops. By changing the amino acids, they altered the physical shape and the electrostatic charge of the hands.
So they changed how it physically feels.
Exactly. They created steric hindrance. That means the new shape physically bumps into and repels native mammalian proteins, but it fits perfectly into its mutated partner. After rigorous testing, they finalized two variants, which they named CX34.7M1 and CX35M1.
The M1 mutants.
Yes, the M1 mutants. And when they ran these M1 mutants through the FETCH assay against native mammalian C by36 and CX43, the flow cytometer showed zero color swapping. Zero. The M1 mutants flawlessly docked with each other, but they were completely invisible to the mammalian proteins. The researchers had successfully engineered a totally exclusive lock-and-key electrical synapse.
Okay, let's just pause and synthesize the sheer scale of what we've established so far with a quick mini recap. Point 1, white perch fish proteins offer a heterotypic, meaning different to different, connection.
Right.
Point 2, FETCH uses red and green color swapping to definitively prove cell docking. And point 3, the M1 mutations created a totally exclusive lock-and-key synapse that ignores mammalian cells.
That's the core of it, yeah.
So, to you listening, think about this. If you were designing a lock that absolutely no existing key in the world should open, how many tiny structural changes do you think you'd have to make? It's atomic level engineering. But a glowing cell in a dish is one thing. Now we have to see if this custom walkie-talkie actually works in a living, breathing creature.
Right, the in vivo validation. And the first one was back in our microscopic worm, C. elegans. They wanted to see if they could permanently alter a behavior. So they installed the M1 pair into a specific sensory circuit.
Which circuit?
The AFD sensory neuron, which detects temperature, and they connected it to the AIY interneuron, which drives movement.
Now, the research notes that doing this, quote, bypasses presynaptic plasticity. For you listening, let's unpack why that is so wildly significant. Normally, a worm has to learn to prefer a certain temperature through chemical changes and repeated exposure. That's plasticity.
Right. It's a dynamic, fluctuating process that takes time. But by installing this permanent, exclusive electrical bridge, the researchers didn't teach the worm, they hardwired it.
Wow.
Yeah, the worms immediately exhibited a persistent behavioral preference for warmer temperatures. The engineered synapse functioned flawlessly, and it completely overrode the natural learning process.
And here's where it gets really interesting, because worms are great, but the ultimate goal here is the mammalian brain.
Yeah.
So they scaled this technology up to the mouse microcircuit.
Yes, and this is where the precision of LinCx really, really shines. They targeted the medial prefrontal cortex of the mouse. This area regulates complex behaviors, and it contains a well-mapped microcircuit consisting of two very distinct cell types.
Okay, what are they?
First, you have excitatory pyramidal neurons, or PYR cells. These are the main drivers of activity. They push the gas pedal. Second, you have fast-spiking inhibitory parvalbumin interneurons, or PV plus cells.
And they are the brakes.
Exactly. They act like the brakes, keeping the whole system balanced.
So the scientists installed the CGuys34.7M1 half into the pyramidal cells and the matching CGuy35M1 half into the PV plus cells.
Correct. But proving that these two specific cell populations were now electrically talking to each other required massive data analysis. They couldn't just look at them. They had to measure the brain's electrophysiology using these high-density silicon probes, specifically looking at local field potentials.
LFPs.
Right. They were searching for a phenomenon called phase amplitude, coupling between two different brain waves.
Okay, let's visualize this for the audience, because brain waves are basically just massive patterns of electrical activity. The pyramidal neurons fire in a slow rhythm called theta oscillations, which is around 4 to 10 hertz. Imagine theta waves as deep, slow, rolling ocean swells.
Oh, I like that.
And then the PV plus cells fire much faster, creating high-frequency oscillations, or HFOs, at 80 to 200 hertz. So imagine these HFOs as rapid ripples on top of the water.
Yes. And phase amplitude coupling means we are looking to see if those rapid ripples, the HFOs, are consistently happening at the exact peak of those slow ocean swells. It's like a conductor ensuring the percussion section hits perfectly on the downbeat of the strings.
If they're locked together temporarily, the cells are intimately communicating.
Exactly. And the data showed a highly significant increase in this coupling in the mice with LinCx.
It did. But they actually zoomed in even further than that. They looked at the single unit cross-correlation in this impossibly tiny window of just 1 to 4 milliseconds.
Yeah, that part is incredible.
They wanted to know, if a pyramidal cell fires, what is the exact probability that the PV plus cell fires just a millisecond later? In control mice, About 11% of the pairs fired within that tiny window. But in the LinCx-edited mice, that number jumped to 17%.
And an increase from 11% to 17% might sound like a tiny statistical bump. But think about the cascading networks in biology. In the brain, a single millisecond is an absolute lifetime.
Right.
And this tiny microscopic tightening of the electrical timing triggered a massive observable behavioral shift. When they tested these specific LinCx-edited mice, they showed a significant increase in social preference.
It's just crazy. Just by physically tightening the electrical coupling between those two specific cell types, they literally made the mice more social.
It is staggering to think about. And for their final validation, they pushed the technology to its physical limits. They went to long-range networks. They wanted to see if they could wire two completely distant brain regions together.
Where did they target?
They put one half of the synthetic synapse in the infralimbic cortex, and the other half deep, deep down in the brain in the medial dorsal thalamus.
I mean, that is a massive anatomical distance to lay a microscopic cable.
It really is. They used optogenetic stimulation, which is shining a tiny pulse of light on the cortex, to prove that the electrical signal instantly traveled down the engineered bridge to the thalamus. And the connection worked perfectly.
And behaviorally, the results were profound here, too. They subjected the mice to a tail suspension test. That is a highly stressful event that usually causes mice to eventually just freeze and give up.
Yeah.
But the LinCx connection completely prevented this stress-induced immobility. The edited mice just kept fighting, kept exploring. It fundamentally altered their stress resilience.
It really did.
So time for a final recap. Point 1, LinCx successfully hardwires temperature preference in worms. Point 2, in mice, it increases that millisecond level coupling between PYR and PV plus cells, the theta and HFO waves. And point 3, this microscopic scaling alters highly complex social and stress behaviors. So, question for you listening. Notice how a 1 millisecond change in microscopic cell coupling cascades into a complex organism deciding to be more social or deciding not to give up under stress. Where else in biology or even in our own lives do we see tiny, almost imperceptible micro-changes creating massive system-wide macro effects?
It's the ultimate butterfly effect mapped right onto neurobiology.
It really is. Which brings us to the broader implications of what we've unpacked today. We've journeyed from a fisheye to a glowing color-swapping cell in a dish, to hardwired worms, to more social stress-resilient mice.
And if we connect all of this to the bigger picture, Lindis represents a true paradigm shift in neuroscience. Previously, our best tools for interrogating these circuits were exogenous.
Meaning they required an outside trigger.
Exactly. Optogenetics requires researchers to physically jam fiber optic cables into the skull to shine lasers. DREAD-Ds, another technique, require injecting specialized designer drugs to activate the cells.
You essentially always need the scientist in the room to flip the switch.
You do. But LinCx is completely endogenous. It uses the brain's natural, existing electrical activity. You build a structural bridge, and then you just step away. The brain drives its own traffic over it permanently. We're no longer just stimulating the connectome, we're structurally editing it.
The sheer scale of this achievement is what continually blows my mind. The effect sizes literally magnified as they moved up the chain of complexity. From the atomic docking of single cells to local prefrontal microcircuits to long-range thalamic brain networks, ultimately manifesting in actual physical complex behavior. The murky, imprecise diagnostic waters of neurobiology we started with. They're finally starting to look like a crystal clear, highly engineered schematic.
It forces us to entirely rethink what might be possible for mapping and eventually repairing dysfunctional circuits in the mammalian brain moving forward. The precision is just unprecedented.
And that leads me to a final provocative thought for you to chew on as we wrap up this deep dive. If we now possess the technological capability to selectively wire any two distinct cell populations together with a permanent, active, highly exclusive electrical bridge, what happens if we decide to get creative?
Oh, wow.
Right. What if we wire a primary sensory cortex directly to a motor cortex, completely bypassing the brain's natural processing centers? Could we eventually engineer completely new synthetic reflexes, or even entirely new senses in mammals? We are no longer just observing the brain's circuitry. We are finally learning how to write our own blueprints.